Good afternoon, everyone, and welcome. Thank you so much for joining us for the first Illuminate webinar from North Star ALS. This is the first of 4 webinars in this series to talk about precision medicine in the context of ALS. And we're also bringing you content on social media for the next 8 weeks and also through podcasts. We really want to drive conversations about more targeted drug development with you. So a few logistical things, as I was saying, North Star ALS is the independent science hub for the ALS community. We're here to connect the ALS community with research in many different ways. This webinar is being recorded and it will be shared afterwards. Thank you so much for your pre-submitted questions. We do read every single one, and we have the ones that are relevant today on hand. Please feel free to drop questions in the question box, and we'll try to get to as many as we can at the end. We will be prioritizing topics that are most relevant to today's discussion. And then go ahead and look out for answers to your questions also through additional content that we're sharing. Some of them were very relevant to some of the other webinars and podcasts that we're doing, so you may see those show up there. We wanted to take a moment and say thank you so much to our sponsors and our community partners who were also very interested in this topic and in bringing it to the community. So thank you so much to all of them. And we have our esteemed list of panelists here. We have Dr. Benatar from University of Miami Miller School of Medicine. He's spent his career changing how we think about neuromuscular disease, from understanding the earliest stages of disease to leading critical work in biomarkers and pre-symptomatic research. His work is reshaping the way clinical trials are designed, and he's brought us closer to understanding when intervention may be most effective. We have Dr. Corey Painter, currently with Even One Ventures, who brings the oncology perspective. Decades of work there have transformed cancer from diseases defined primarily by where they started in the body to diseases increasingly understood by their underlying biology. She's been a leader in translating science into progress while keeping patients at the center of every decision, and she understands it well because of her own lived experience with cancer. We have Dr. Juliana Acosta Uribe, who's dedicated her career to working with families affected by dementia and neurodegenerative disease in South America and bringing their samples and experiences back to the lab across continents to UCSB. Her work reminds us that understanding disease biology is better when we understand the impact on people and families living with these diseases. We have Dr. Joe Lukock, who has spent years bridging neuroscience, drug discovery, and clinical development, helping move potential therapies from the lab into clinical trials. He is now Chief Scientific Officer at Denali Therapeutics. His perspective spans the scientific challenges and the practical realities of developing new treatments, and that makes him really well positioned to have this discussion today. What also makes this panel special is that each of these individuals has dedicated their career not only to advancing science, but to partnering with the communities they serve. They understand that every sample, given every biomarker study, every clinical trial, and every family that chooses to participate in research contributes to advancing the field. Thank you so much, all of you, for being here today. And let's go ahead and get started. I'm going to stop screen sharing. All right, so we have four main topics today. So the first topic is how do we get to better treatments? And I'm going to start with you, Michael. We hear terms like precision medicine and targeted therapeutics all the time. If we put the terminology aside and focus on the goal, which is developing treatments that are more impactful for people living with ALS and FTD, what are we still missing, and how much of the biological diversity of these diseases do we actually understand today?
Yeah, thanks, Nadia. Great to be with you in this panel. I mean, I think we're still really missing is a basic understanding of what the pathophysiological drivers of disease are. In the genetic forms of disease, we understand the upstream causes. I mean, people where we don't know the gene, we fundamentally don't understand what's driving the disease. And so we try to get a handle on what mechanisms might be at play. But often we're looking at sort of downstream mechanisms that may be a consequence rather than a cause. And what we really lack is the biomarkers to help tell us what's the biology that's driving the disease. And without that knowledge, it's really, really hard to sort of think rationally about drug development. We don't know whether we're going after the chicken or the egg. I'm not sure which in this analogy, but the point is we don't know whether what we're targeting is an epiphenomenon or a downstream problem as opposed to a driver of the disease. Just a word about sort of biological diversity. I think right now, to some extent, this is a hypothesis that we still need to test. What we know is that most people with ALS, about half of people with FTD, share the biology of TDP43 pathology. There may be different upstream mechanisms that can lead to that. There may be multiple downstream consequences. But whether we need different therapeutics for people who all have the same core underlying pathology, I think is a hypothesis that we don't yet have an answer to, mostly because we lack the tools to be able to discern that.
Thank you so much for that response. And Corey, in cancer, one of the biggest shifts was recognizing that diseases we once grouped together were biologically distinct. So, what changed when researchers started looking at biology rather than diagnosis alone in oncology?
It's a great question, and I'll talk about this in terms of two of the big cancers, so breast cancer and lung cancer, for example. Those were just one entity at one point in time, and they were all treated the same with chemotherapeutic agents, but people would have vastly different responses to the same types of drugs. And I think that made people try to dig into the underlying biological causes. And it led to differences in how these two diseases started to become parsed based on the underlying biology. And as soon as the biology was understood, it made all the sense in the world why a ER positive breast cancer patient might not necessarily have the same response as a HER2 positive breast cancer patient or a triple negative breast cancer patient to the same exact therapy. So I think it started with a recognition that patients, there's a diversity and a heterogeneity within a particular disease. And once people started to try and understand the biological underpinnings, they were able to map some of those to the outcomes that they were seeing. And then conversely, loop back in order to try to tailor drugs specifically for these subtypes of patients. In breast cancer, this was more a result of looking at protein and RNA expression. And so you see differences in the expression of transcription factors like estrogen receptor and or HER2, and also the absence of both of those, which give you triple negative. Those are three distinct diseases with vastly, completely different treatment approaches. That was never appreciated until we started to understand the underlying biology. If you go over here to lung cancer, there was once just lung cancer. And then histologically, you could see that there were kind of two types of lung cancer by the way they looked and acted. One was small cell lung cancer and one was non-small cell lung cancer. So just a bifurcation of the way that they were classifying these two types of lung cancer. And they had very different clinical courses. And so that was just sort of like one recognition. But then you start layering in the underlying genomics. And within non-small lung cancer, there are these small populations of people, sometimes only 1%, but they have a very distinct driver that we know is causing their non-small cell lung cancer, which is a little bit different than what Dr. Benatar was saying in terms of we don't know what's driving versus upstream, downstream, and how to target it. We knew in these cases that there is a ROS1 translocation, and that is going to drive this disease. And there is an inhibitor towards that thing, and you can put somebody into a durable, have a durable response as a result of treating that one thing we know is the driver. So in a lot of cases, it just had, to try to make a more pithy answer, understanding the biology led to our ability to target different approaches, therapeutic interventions.
Thank you so much, Corey. And Michael, I wonder if you have anything to add to your response. So I know that we hear a lot that the TDP43 is the underlying biology. And so is there anything there? It sounds like there's more distinction in cancer, in different types of cancer.
Yeah, I mean, I think it's an area that we don't understand well. If we start with the genetics, we know that SOD1 ALS is different to non-SOD1 ALS, and we wouldn't bring sort of an SOD1 lowering therapy to people who don't have SOD1 ALS. So I absolutely endorse and agree with the idea that there are different biologies and that we'll need different therapeutics to target that biology. What we don't yet know is what that biological diversity is in people who share the same common neuropathology. And are there different drivers of that? If there are, or if there are different downstream consequences or secondary drivers, then we may well need different therapeutics. But because we don't have the tools to answer those questions, that remains a hypothesis, I think, at this stage for the ALS world.
That really helps to hear. And Juliana, where do you see the biggest gaps between what families are experiencing and what researchers are currently understanding about the disease biology?
Well, I would say that there are two parts of that question. One of them is what families believe that researchers still do not get. And then the other thing is what families believe it's their role and what they are gaining out of the development of novel therapies. So, in terms of the families that I've worked with, I have noticed how they're very conscious on all the little differences that happen between individuals of the same individuals who carry mutations in the same gene or individuals who carry mutations in different genes. For example, I've been working with a cohort of patients with SOD1 mutations in Colombia, and it's really interesting how they view the illness very differently because some of these mutations, even if they're in the same gene, have a slowly progressive ALS for which the carriers say these novel ASO therapies do not offer us anything that's worth pursuing. I don't care if they bring it to the country or not. I think the downside is far worse than the effect of the therapy. However, even other families that have mutations in the same gene will have a real quickly evolution of the illness. And then they say, no, we do want that medication. So families have very different points of view of the use of this personalized targets. And something that I've noticed that is a constant between families within the ALS, neurodegeneration, or FTD, or Alzheimer's disease is that when they're participating in studies trying to deliver or develop personalized diagnosis or treatment for genetic forms of this diseases, sometimes they worry if these medications get approved, will they have access to them? Will the families keep having access to these different medications? And the social, personal experiences of those living with the illness and participating in trials, saying, how will this help me and my family, not in the future? Science is promising, but in the right now. So the need for engagement and social programs that are tied to the research in which the families can have intermediate and short-term benefit from participating in any kind of medical research, even if it's non-pharmacological or clinical trials.
Thanks for sharing that. Those additional studies, we've had this conversation, Dr. Benatar and I, in the podcast episode that matches this part of our series that talks about participation in other types of studies that are driving the research that makes all of the other research go faster. So, Joe, from a drug development perspective, do you think the future in ALS FTD are a few broadly effective therapies, many targeted therapies, or some combination of both?
Yeah, excellent question. And first, I want to start off. I mean, to echo what Juliana just mentioned, just to thank every person living with ALS who participates in a study. It's absolutely critical to what we do, and I know that it's effort for anyone who participates, but the drugs don't move forward without that. So just to say that, just to start. So from my personal perspective, I've spent 20 years plus trying to develop therapies for ALS. And for a long time, my hope along the lines of what Michael was saying, is that, there's going to be some common thread that ties together individuals living with ALS. And there's going to be then the opportunity to use those mechanisms to develop therapies that are going to be broadly effective. I would love to say that that is going to work out eventually. I will say thus far, that's been difficult, and we haven't had a lot of successes as a field. And it boils down to really what Michael was saying, which is we have a pretty poor understanding of the biology that drives ALS. Despite many years of research, we still have a lot of work to do there. TDP-43 is an anchor point, but we need others. And so to really draw in the oncology analogy, I do think that's where we're most likely to have near-term success and where we have had success, which is stratifying patients based on a particular molecular driver. And I think that there's a reason why the oncology field has made such progress, and they've done just that. And so for things like tofersen as a treatment for SOD1 driven ALS, we've seen, I think, what is our first glimmer of hope that we can really impact this disease in a positive way. I think that's held true in, some data rolling out for FUS-driven ALS. So for these rare genetic forms of disease, I think we're starting to make progress. The challenge for us as a field now is, you know, about 10% of people living with ALS have a known genetic factor. The other 90% don't. And so how do you stratify those patients? How do you define what those mechanisms are so that we can go after them? And that's what we're spending a lot of time doing now. I think that there's more tractability there, and that's the hope, but there's work to do. And so, one of the challenges, and I think, Juliana mentioned this with SOD1, which is, when you're living with ALS, there's different courses of disease. There's different, some people progress more rapidly than others. Some people have different sites of onset. The challenge is the molecular correlates, the biology underlying it, isn't shared with those different metrics. It's all random across the board. So you can have someone with an SOD1 mutation who progresses slowly or quickly, right? So it doesn't give you a hint in terms of breaking up that patient population. And so I think there's a work to do there, but it's a long answer to the question of, I really think that finding these patient subtypes and then really being able to say, let's develop an effective therapy for that group of people, is where I think we have the most opportunity to develop effective treatments for ALS.
Thank you so much for that very detailed response. So we're going to dive a little bit more into the oncology analogies and what translates and what doesn't. And Corey, you already gave us a little bit of an overview about the science, but can you talk a little bit more, looking back? You've been here for some time. What scientific, clinical, and cultural shifts had to happen before precision approaches started delivering more meaningful results?
Yeah. I'll also try to couch this in what, obviously being very naive to the whole world of ALS research. So please don't take anything I say if it's out of context. But I think that the number one thing that is needed is models of the disease. And I don't know where the state of ALS research is in terms of modeling. If you can get people's skin or muscle cells and derive iPSCs from people that have fast progressing versus slow progressing, even if they have that same variant in that same gene, but then ask what else is going on that might contribute to a polygenic risk score for progression. So you could develop kind of like prognostic tools based on people's biology, but like just being able to model the disease, I think is very critical. And I don't know how you do that with an entire, system where you're trying to understand what the underlying biology or the genetics that kind of contribute to that are. But being able to model the disease and then, being able to have a multimodal approach to understanding biology. So in cancer, it's not enough to just know what the mutations are because you don't know what the driver is if you only know what a mutation is. So you have a bunch of variants of unknown significance, and that's sort of where it starts. Like, what is the role of this gene that we know is mutated in this subpopulation of people? We know it's a problem, but again, it sort of reminds me of what I'm hearing here. We just don't know. Is it a driver? Is it upstream, downstream, and what is really going on? So you start with one type of modality and understanding what's different in the biology, but then you have to layer on multiple different types of information. So you need not just what is the thing in the DNA level, but what is going on with the transcriptome. So how are the genes being expressed differently in these subpopulations of people? But starting from the perspective of, I know this population of patients is progressing faster than this population with the same underlying gene. What else is different at the RNA level, for example? And then a step above that, what's going on in the epigenome? Are there differences in the way the chromatin is opening in this set of patients here that are, that's different than this set of patients here? So, and then pulling all of the information together to have a comprehensive multimodal understanding, but it has to be baked in clinical outcomes. So you have to be able to start with populations and cohorts where what the outcomes are, but you've collected all of the biology and the data, and you can go back and kind of retrofit what you're seeing in what actually ultimately happens with the outcomes.
Nadia, can I jump in with a question?
Yes.
Just to have a discussion. Corey, I wonder if you could sort of comment, and forgive my ignorance in the cancer world, but the enormous advantage you have is that you can biopsy the tumor or take it out and study it. We simply can't do that in ALS. And I wonder whether you could comment on the extent to which cancer could have made this progress if you didn't have access in that way.
It wouldn't have. Yeah, and that's why I led with, could you generate iPSCs, for example? Could you model the disease? I think you have to start there. You have to be able to model the disease in some way, but I also think you could get hints and clues with the technologies that we have that have been built off of the successes in cancer and translate them to this disease. So collect CSF, collect blood, collect serum, collect plasma, do imaging, generate patient level individual models of their own genetics, and then layer on what you're seeing in terms of how that alters the mitochondria in those particular neurons that you've modeled from that human's DNA iPSC. So I think you couldn't start there de novo, but I think there's a ton of lessons learned and technologies developed that you could co-opt. And then once you stratify the patients based on outcomes, go back and ask what's different in any soluble markers, and or what can I see when I model from an individual patient's cells that I could then learn about why is this population with SOD1 variant progressing, and this with the exact same SOD1 variant not progressing? What else is contributing to the polygenic risk that I can now see because I have different models of the disease? That's what I think you could do now, at least, but you couldn't have done it where cancer had an advantage from being biopsiable.
Thank you so much, both of you, for that. So yeah, we're seeing this clinically, I think, in SOD1 and in talking with families, the heterogeneity even within that one gene and the response to tofersen. Michael, do you have anything to add to follow up? Okay. Joe, hearing what Corey is sharing with us, and you talked a little bit about the nuances in cancer versus ALS. Can you do a little bit of a deeper dive? Where are the places where there may be overlap, and where are we fundamentally different?
Yeah, I think that there's a lot of potential overlap there. I think that, Corey has it just right. And what she mentioned as well, which is also correct, is, you can't just go do a brain biopsy the same way that you could biopsy a tumor. And that's part of the challenge, right? And this is where, the work that Michael has been doing, and we've worked with him for many years, around collecting CSF, collecting biomarkers, trying to have some other way to understand what the biology underlying disease in a particular patient is. I think that's going to be hugely informative. The trick is it's a lot more challenging when you're trying to do that for someone's central nervous system versus a tumor of any kind. And so it's going to take us a bit longer to get there, but an effort that's very worth taking, right? And so I think it's those kind of things combined with other things like imaging. I think that's going to begin to allow us to define those biologies and those groups of individuals with different types of disease, so that we can start to develop those therapies. And what's the limiting thing for us right now in terms of therapeutic development is we don't quite have enough information to say this is the therapeutic we want to develop yet. And we're getting close, but not quite there. And so we take our best shots at it, but I think a little more information would be great. I will also say, like, a word from, like, this is not impossible. There are other diseases that are related. And I'll give an example of Alzheimer's disease, where a lot of this work is also being done. And there's now biomarkers. There's a blood-based biomarker that can tell you whether or not you have Alzheimer's disease. There's still no marker that says you definitively have ALS. And, that's a big challenge in and of itself, right? But I think we're making progress towards those things, and they're going to have a tremendous impact in terms of therapeutic development.
And that was a great segue into talking a little bit more about biomarkers. So, Michael, we have talked a lot about one of the biggest challenging is, are we measuring and changing the right things? So, what can biomarkers tell us today about disease activity, progression, and treatment response, and where are the gaps that you're seeing?
Yeah. So how long do I have? No, I'll try to be brief. So you asked a couple of questions in one there. You sort of alluded to disease activity, progression, treatment response, and I think what lies behind that question is the idea that biomarkers can serve different purposes. We have a separate podcast about that where we've sort of delved into that, and we'll encourage people to listen. But maybe I can say a word about neurofilament light as what I think is the current biomarker that is our most powerful tool. So, what does this tell us and what doesn't it tell us? So, what neurofilament tells us is about the aggressivity of disease. So, higher levels mean the disease is progressing more quickly, lower levels mean the disease is progressing more slowly. But for an individual person, neurofilament levels are relatively stable over time. So it's not, for example, capturing disease progression. Disease progresses, people get functionally worse, but neurofilament levels stay the same. So that's just sort of an illustration of how the same marker or a marker can tell us one thing, but not another. But that stability is enormously helpful because then when we give a drug like tofersen and neurofilament goes down by 50, 60%, that tells us that we've exerted a biologically important effect. And then when that translates into something that's clinically meaningful that matters to people with disease, that sort of ups the ante and tells us even more so that's an important tool for us to now use in drug development. So that lesson, getting our foot in the door, tells us we can use this as a tool for future drug development. It's not the be all and the end all. There are limitations to our knowledge. There are caveats. We don't understand, for example, what are some of the mechanisms responsible for clearance of neurofilament and what might lead to levels being maintained at an elevated level. So there are things we don't know, but perfection shouldn't be the enemy of the good. It's a tool we understand well and we can use. But it is a marker of downstream biology. It's a marker of neurodegeneration, of axonal loss. It is not specific to ALS. It's not specific to any one type of ALS. And it doesn't at all mitigate the need for us to have biomarkers that tell us about that upstream biology that could help drive our thinking about what are the targets we want to go after and which are the subset of patients that might then potentially benefit from targeting that particular mechanism.
Thank you so much, Michael. And just building on that a little bit, your work has focused on people with genetics for neurodegenerative disease before symptoms begin, as well as those living with the disease. So maybe you could speak a little bit about the context of NFL in the pre-symptomatic space. How might that influence therapies we develop in the future?
Yes, you're referring to our pre-symptomatic familial ALS or pre-FAL study, where we've been following people at elevated genetic risk for ALS. One of our key observations, it's hard to believe almost a decade ago, was that neurofilament goes up in the blood of people with rapidly progressive disease. This matters because neurofilament represents the speed with which disease is emerging. And so in that subset of genetic carriers, we see an early pre-symptomatic rise in neurofilament levels. And that information was key to us then working with Biogen in the design of Atlas. where we're using tofersen as an SOD1 lowering therapy pre-symptomatically based on a rise of NFL to see if it's possible to delay the emergence of symptoms of disease or possibly even to prevent it. But we've now since built on that, and this work is on preprint. It'll be out hopefully soon. But using a proteomic approach to look at thousands of proteins and to map the trajectory of these proteins and to see that we can see other proteins that are changing not just in a year or two before people develop symptoms of disease, but in the decade before people develop symptoms of disease. And so I think these now represent an important new emerging tool that will help us think about what are some of the biological drivers of disease and what are some of the opportunities to intervene therapeutically earlier. And before I stop, just to tie into something Joe was saying, when we study biofluids in people who've already got disease, they've got manifestations of disease, we don't know whether that's a cause or effect of the disease. When we're studying people before disease appears over the clinical horizon, at least we have temporality on our side. So if we observe biomarker A go up before biomarker B, we don't know that that's cause and effect, but at least it's plausible that that biology that we see changing early is a driver of what comes later. So it takes us a step incrementally forward in terms of trying to tease apart what's the early biology and what's driving disease. So I'll pause there.
Amazing. And Joe, from the industry side, can you talk a little bit about how biomarkers influence which therapies move forward and how they move forward? When a trial fails, how do we know if it's the drug or the inability to identify the right patients or the right outcomes?
Yeah, it's a great question. And maybe to play on that, I'll also touch on what one of the questions was in the chat from Kevin, I believe, who was asking about. So we recently ran a trial at Denali that unfortunately failed in ALS. And the compound was called DNL 343. And, I think the question online was, are we going to share information? What can we learn about that study? And what I'll say is what we want to do as drug developers is ideally we make drugs that really help patients. But even if we don't, we want to make sure that we learn something from running that clinical trial and we understand a little bit more about disease. And so our DNL 343 study was designed to go after that TDP43 that Michael mentioned. So in a dish, when we grew neurons in a dish, DNL 343 impacted the aggregation of TDP43. So we were hopeful that this would then have a positive impact for patients. So we developed the drug, we ran a clinical study, and what you ideally want to do in those clinical studies is the first thing you want to measure is, can I impact TDP43 in a patient? Is that actually happening? Yes or no, right? And if the answer is yes, it's impacting it, then I can say, okay, well, if I'm impacting that biology, does that have a positive impact on disease? Yes or no? And in some ways, regardless of what that answer is, you learn something, right? So if it's yes, we have impacted this biology, no, it didn't impact disease, that helps us learn as developers to say, okay, this is not a relevant pathway. We need to move over here and try these different things. And so that's what we try to do. I will say that's not always the way clinical trials have worked, unfortunately. And I think part of the reason why is there are not very many biomarkers. So Michael mentioned NFL, which is a very useful biomarker, but it's not specific to any one type of ALS versus another type of ALS. And we need to start increasing the number of markers we can measure in people when they're enrolled in the study to understand what's happening, how it's impacting biology. And so we very much want to do that. We've shared some data from that trial. We want to share more data as we're continuing to gather and learn. But I think the more understanding of biology, the more things we can measure, then the more we're going to learn for each clinical trial and the closer we're going to get to what I believe is going to be really truly effective therapies. It's going to, I hope we're getting closer, and it's those learnings that are going to take us there.
Thank you so much for your very detailed response and for addressing Kevin's question. Um, we're going to shift a little bit to talk about representation in research. So, Juliana, much of our understanding of genetics has been built from populations of European ancestry, and yet ALS and FTD affect people around the world. Can you talk about your work with families in South America and what we're missing when large portions of the global population are underrepresented?
Thank you, Nadia. This is one of my favorite stories. About 5 years ago, we were doing a systematic analysis of a cohort in Colombia. We recruited 1,000 individuals with different forms of familiar neurodegeneration. Some had Alzheimer's, others had early onset dementia that had not been properly diagnosed, ALS, FTD. So we were doing a very systematic search for genetic causes. Then we were looking both at their ancestries, both in a global mm, way, like how much do you have of European, Amerindian, African, and also which segment of your genome came from this particular ancestries. So after doing those examination for the genes that I was taking from OMIM, which is the Online Mendelian Inheritance and Men database, I took the set of genes, looked for Alzheimer's disease. Then Alzheimer's disease was very I found a lot of mutations of different origins that were very similar proportion in as to the global ancestry of the population. So I kept moving. I looked for frontotemporal dementia, and I got about 6 or 7 families with genetic FTD. When I looked at the local ancestry of each of these mutations, I was, oh, interesting. They're all of European origin. But I didn't give much thought to it as I kept going. And then I looked for, okay, my next pool of genes is going to be associated with ALS. And then I was hoping that some of the families that we had with ALS were going to pop up there. But I saw a lot of variants in genes that I was pulling from OMIM that were associated with ALS, not FTD, but in my cases and in the families that I was seeing, they were manifesting as FTD without any motor neuron involvement. Surprising variants such as some in SOD1 we're seeing in more than one case with frontotemporal dementia without motor neuron involvement. What was the most surprising is that the haplotype, which is the chunk of DNA in which this mutation resided, was of Amerindian ancestor, which meant that the individual had inherited this specific mutation from an Amerindian ancestor generations ago. Then is what it hit me that I was trying to look at these illnesses with what I call the European paradigm of knowledge, where all the searches that we're doing of genes, causal variants are somehow being based on previous studies being done in European populations. This doesn't only affect when we're looking for monogenic causes for ALS, it will also change the way we interpret polygenic risk scores. How polygenic risk scores, they work beautifully when predicting any X illness in European population. But as soon as you moved it to a new population, the power for detection definitely went down. And as we keep trying to include more and more populations in diversity studies, we're seeing that there are holes that we need to fill. That if we keep working the same approach in the same population, it's just like if you have a dice and you're only looking at it from a single approach. So it is, I'm going to say, mandatory to include people from different backgrounds so we do not focus on single genes or single approaches. We really need to go forward and see where do all of these different, and I'm going to use the term genetic burden, which is how the genetics of each population come together. Where do they all meet? What do all the populations have in common that we can see are the drivers of the illness beyond what we call population stratification?
Thank you so much for that. And Michael, how do you what impact do you see from this in the natural history studies that are ongoing today? Can we build targeted approaches if our data sets aren't reflecting that broader population?
Yeah, I mean, I think it's always the case that if we want to answer a particular question, we have to be sure that the resource we're using to answer that question is up to the task. And so I think it depends on the question, right? And in my mind, there's no question that we're trying to understand, for example, genetic contributions to disease. I think Juliana's right. We've sort of only been looking in one place. And we need to look in other places to understand the genetic contributions to disease in other populations. What I think is an open question, and we'll only answer this by making sure that we embrace sort of the full biological diversity of disease as we study therapeutics, is to what extent, for example, is there a pharmacogenomic influence on therapeutic response? So, to what extent does your genetic background or a polymorphism in a particular gene impact therapeutic response? A critically important question, but we have to have therapies that work or that show some promise if we want to be able to interrogate that. But I think there's absolutely no question that we undermine our efforts if we sort of focus our search too narrowly.
I appreciate that. I also appreciate your shout out to pharmacogenomics. I think we need to do some social media content on that. I've spent a lot of time with that in the last few years. All right, we're going to shift to, oh, go ahead.
No, at some stage, maybe call on me to answer Kevin's question in the chat, but I don't want to disrupt our flow.
Oh, go for it. Go ahead and answer, and then we'll come back to the last main topic.
Sure. So, Kevin, just very quickly, I think that we have to understand how we're using the tool and for what purpose. And so I think you're right. If we have a drug that's targeting a mechanism of action where we don't expect, for example, to see a change or an impact on axonal degeneration, then neurofilament might not be a great tool to measure response. But where I would say it is still a good tool, because it is also prognostic, is when we're doing a small phase 2 study and there, just by its nature, because it's small, even if randomized, group imbalances in how quickly disease would progress between the treated and the placebo group, we can use neurofilament as a prognostic marker to adjust at baseline for some of those imbalances and help give us greater confidence that if we're seeing a therapeutic effect, or an apparent therapeutic effect, we have a greater confidence that we can attribute that to the drug and not to natural variability. So, in the podcast Nadia and I recorded, I think we spoke about context of use a lot. Maybe it's become one of my favorite phrases. But what I mean is we always have to make sure that we're using the tool for the right purpose. And so understanding how we're using the biomarker tool and using it correctly will help make sure that we derive benefit from it and don't get misled.
And I just want to add Kevin's question, in case somebody doesn't have access to the chat, is are we putting too much emphasis on NFL and trial design? For example, if UNC13A ASOs work to strengthen synapses but don't repair or protect axons, is expecting a change in NFL realistic? And so it sounds like it comes down to that context of use on how NFL is used. So thank you so much, Kevin, for your questions. All right, last topic here for everyone, and then we'll jump to some more questions. What needs to change? So, Joe, if and I hate questions like this because I'm going to ask you the single most important thing, and there's a lot of important things. But if we're serious about developing therapies that have a bigger impact on the diseases, what scientific or strategic gap needs to be addressed?
Yeah, I think we've touched on it a bit today. I think we need to make a real investment in understanding the different types of biologies that can drive disease. And what I feel like happens with ALS a little bit too much is there's the genetic populations that are easy to identify, and past that, there's maybe more effort needs to be made to really say, like, let's not just lump them all together. Let's really figure out what's going on. And I think that that's going to give us higher potential to have effective treatments. And again, I think we're making progress. Great science being done. I just would implore the field and everyone to get involved in that as much as we can. And if we have a better understanding of disease, I promise we'll work very hard to leverage that to make good therapeutics.
Thank you. And Corey, cancer research has shown so much promise and also some of the limitations of targeted medicine. So looking back, what's a mistake that we might be able to avoid, and what's one investment that might pay off over the next decade?
I don't know that I could limit it to one mistake. We're still making mistakes, to this day. I do want to maybe highlight. Let me think more carefully. Is there a mistake you could avoid? I actually would call out what Juliana was saying, in that we need to have representation. That's the number one biggest mistake I think we've made in cancer. And it's cultural. It's also gender, and so I think we have to have good representation. Full stop. That's a big mistake we made. Moving forward, I just want to also highlight innovation and the role that innovation can play as this field tries to grapple with how do you get biology when you cannot do a biopsy. And I'll use a vignette from my job that I had before my current job, where I built a liquid biopsy company where we profiled circulating chromatin from plasma. And we struck up a collaboration with a woman who she was a brain physiologist studying multiple sclerosis in Belgium. And she was really trying to benchmark anything we could see in the plasma to, anything she could see by PET scan of the rim around the lesions and how active that rim was in multiple sclerosis. And so we went ahead and started profiling plasma from healthy and relapsing, remittent, and different people with different stages of the disease. And it was shocking how much we could see in the plasma of people with MS. Shocking, like what we could see in circulating DNA that was coming from the brain, but we could still see it in the plasma. And I presented a poster at ECTRIMS, and people were shocked by what we were able to see. And we're not experts in this field. And so it would also be, and neurofilament is also a big biomarker in that field as well. And so it's just one tiny little bookmark, but know that that's out there. So you could start to talk about what else could you see from the plasma that is representative of things that actually are coming from neurons that are at various stages of dying. You can see what's dying that's wrapping around a neuron. You can see what's dying from the neuron itself. And some of that actually does populate the plasma ctDNA. And what we were looking at was just an enrichment of circulating chromatin from those cells, and could map back to the deeper biology, not just yes, no binary, but what exactly was contributing to that signal. So I'll put that out there as a potential forward-leaning way of using biomarkers in this disease.
Thank you so much, Corey. And Juliana, similar question for you. What's one investment or change that you think would accelerate our understanding of disease biology and ensure that future therapies are benefiting more people around the world?
I think we need to improve in the communication between the FTD field and the ALS field. I've seen this as I approach families with either or, that those individuals that have been initially being assessed by a cognitive group, they have FTD, they're not fully being assessed for ALS, and that's sometimes vice versa, that individuals with ALS are not getting an appropriate cognitive screening beyond the ECAS. We really need to work on this because there is much more in the in between than those two distinct and sometimes meeting in the middle pathologies.
Thank you so much. And Michael, when you think about the next 5 years, what would meaningful progress look like to you? What needs to happen to move us from talking about better therapeutics to actually delivering them?
Yeah, so thanks for the question and lots of thoughts. I'm going to start with just by picking up from Juliana. We long felt that, we're too siloed as communities. Um, we haven't said this, so I'll say it, that ALS due to TDP pathology and FTD due to TDP pathology is the same disease biologically. They just have a different phenotypic expression of the disease. And a therapy that's likely to work for one is likely to work for the other. So this comes back to the idea that when we're thinking about developing therapeutic paradigms, we really have to think about understanding the biology and targeting the biology. So I'm taking us back to where we began. We need biomarkers that tell us about the pathobiological drivers of disease so that we can target that biology at some level, irrespective of the phenotype. And that takes me to the next area where I really hope we'll move. We've begun to enter this era, but I'm hoping we'll move more so into the era of earlier therapeutic intervention and maybe even pre-symptomatic therapeutic intervention. We know this from cancer in spades, right? I think I'm not an oncologist, but if you treat cancer early, you're more likely to be effective than if you treat it late. Maybe that's a given in every disease, but we treat neurodegenerative diseases and ALS relatively late in the course of disease. Again, biomarkers can help us think about earlier diagnosis, earlier therapeutic intervention, maybe even pre-symptomatic intervention and disease prevention. We don't know whether we're tossing out perfectly good therapies because we're giving them to people too late in the course of disease. So I think there are multiple layers here, understanding biology, intervening therapeutically at an earlier stage. But maybe the last thought I want to share is that I think we need an ecosystem of therapy development that's a learning system. And Joe mentioned this. We need to learn from every trial and every study, whether it's quote unquote positive or negative. We have to learn. We have to avoid repeating the same mistakes over and over. And ultimately, we need for our trials to be smarter, to be more efficient. And then these are more likely to deliver on the promise of effective therapies. But to the extent that we keep sort of doing the same thing over and over because it's what we've done before, I think, well, let me just say, I hope that never mind 5 years, but in the coming 6 months, next month, now, that we learn from what we've done in the past and we move forward learning from everything that we do.
Thank you so much. And we have another question in chat that may build on some of the things that you were just talking about. So Lori Heller is asking, will we ever get to ALS cures with our current clinical trial structure? And if not, how should clinical trials be changed and why isn't it happening? So maybe, Michael, we can start with you on that, and Joe might have something to add there.
It's a difficult question. I do worry that currently, let me start with the good news. The good news is when we understand the cause of disease and we can target it, we know that this disease is actually treatable. SOD1 ALS and tofersen help tell us this. Hopefully Atlas will show us that if we intervene early enough, we can actually really preserve function and maybe prevent disease. So at some level, I think fundamentally the disease is treatable. We just have to target it with the right therapeutic approach and early enough. But I do think that there are things that we are doing that systematically need to change. I think we can't continue to do small, early, and mid-phase clinical trials using underpowered outcome measures like the ALS FRSR or survival. We have to focus, we have to answer the right question at the right stage of therapy development. And in early and mid-phase development, we have to ask, did we engage the target? And did we impact biology? And maybe, do we move the clinical needle in the right direction? But if we try to answer early and mid-phase trial questions or drug development questions by answering efficacy questions, I think we're going to continue to fail. So I do think that that's one thing that urgently needs to change.
Thank you so much. And Joe, what are you seeing what are maybe some of the issues we've had with trial designs and how we should address them moving forward?
Yeah, I think Michael said it very well. I think great points, Michael. And you know what people tend to do is to try to read too much into trials that are not designed to tell them that. And the ALS FRS, you know, it has its issues, but it's a pretty good metric if you do a large enough patient population and you follow them a long time. You can tell whether things are working. But going back to some of the things we spoke about before, and really what Michael mentioned, if you said, okay, the only ALS therapies that get to the large study are the ones that have clear target engagement and they show they're impacting the biology first. If you made that cutoff, very few programs would move past that because people rarely show that, right? So they rarely do the more simple de-risking steps that allow you to move only the best ideas into those big studies. And I think that's the way we need to think about it differently. And if you go to like the oncology field or other fields, that's what they do. They do an early clinical study. You see this evidence of activity very early on, and you can understand it. And then you can design a larger study to really confirm it and go into more detail. And I'd like to see ALS clinical trials move more in that direction as well.
Thank you so much. All right. We have a question from Kevin Robinson for Juliana. How are we integrating other factors like environment into genetic analysis?
Well, I think we're still trying to figure that out. There are so many different studies in which we're looking at the natural evolution of the illness, both for individuals with, let's say, monogenic ALS versus polygenic ALS with an environmental contribution. We're still trying to learn on that. I don't think there has been any significant conclusion yet. We find, I don't know, I'm going to defer this to Dr. Benatar, who's been around the block for longer than I have. So, do you have any input?
I mean, I think the honest answer is that we understand the role of the environment much less well than we understand the role of genetics. In large part, I think this is because we have the tools to study the genome. We have very sophisticated tools. As I often say when I talk to patients in clinic, if I asked you how much lead you were exposed to last year or what your lifetime pesticide exposure was, you'd probably say you have no idea, and that's true for most of us. And so we need better tools, but we also have bias in the way we do these studies. When we ask people about environmental exposures once they've already developed disease, it's human nature to think about what it was that could have been responsible for my disease and to sort of point to it. And so I think we need in parallel, and we need all of this. Think of that movie, What's It Everywhere, Everything All at Once. We need it all at the same time. We need the phenotypic data to understand how disease evolves. We need the whole genome to understand the genetic influences. We need the environmental exposures to understand how they play a role and how they interface with genetic risk. And we need the biofluids to be able to study the biology. And we have studies in our field that are trying to do this, but we need more.
Michael, do you think that comes back to having people participate in some of these observational and natural history studies that we have ongoing?
I do. I think, and you and I spoke about this, I think, on the podcast. We want everybody participating in a clinical trial if they want to and if they can. But there is such enormous value in participating in observational studies, genetic studies, biomarker studies, environmental exposure studies. If we're ever going to get to being able to do more rationally informed drug development where we understand the biology that we're targeting, we have a dearth of information and knowledge about some of these contributors to the biological drivers of disease. And so until such time as we make that progress, we're going to continue to struggle. So I know that there's an allure of our field doing more and more clinical trials. And I often joke that it's not, I say this in jest, I don't necessarily want more trials, I want better trials. I want trials that are better informed by the biology, using the right outcome measures that will give us answers that will help us move the field forward. I don't want us just doing more and more clinical trials that are not informed by biology, that sort of give us the sort of the short-term hope that this will be the drug that works, but don't actually move the needle forward. So we need all of this in parallel, but I guess I want to put a plug in for participating in as many observational studies as possible in addition to clinical trials to help us understand the biology of the disease and the drivers of disease so that we can do more clinical trials that are more likely to yield a beneficial therapy.
Thank you so much for that. Corey, we had a pre-submitted question for you. So you've done some incredible work advocating for cancer after all you've experienced and seen. What advice do you have for us on the patient side on how to advocate for better research?
Thank you for the question. I would kind of echo what I've heard surfaced here today, in that patients have the ultimate control here in what moves forward, and they can both participate in trials and participate in all of the trials they possibly can. That's the number one thing I think patients can do. I think patients can self organize and help fund additional studies. I cannot underscore what I think is the most important thing that I'm hearing, which is more natural history studies with more collections, so that you can really dive into what you're seeing stratified by outcomes. And then, like I was saying before, go back and look what was different in all of the biospecimens that you were able to collect. The only way any of that is ever going to be possible is if patients unite against this disease. And there is a call to action that they can march along to, and so I think it's more of a collaboration between patients and the research community than anything else, and they go hand in glove. They go together, but it 100% requires people that are dealing with this disease. And I don't even just say patients, I mean caregivers and loved ones that are usually the people that are advocating for patients. At least that's what I've seen in my community, to really step up and make sure that the research community has every single thing at the ready that they possibly can: biospecimens, consenting to allow research, and financial contributions to get it done. It is so unfortunate that the burden falls on us as patients, but it does. We cannot wait for other people to come rescue us. We have to actually step up and do a lot of the work ourselves.
That goes straight to my heart. I say that all the time. Well, my dear friend Allison, who's on as a guest right now, always says, "Without patients, there is no research," and yeah, and nobody is coming to rescue us. I definitely I've felt that for the last 7 years. So thank you for your perspective. Um, all right, we have a couple of other questions. Joe, we have one for you that was also pre-submitted. This is kind of a tough question, but we're seeing more and more trials now that are not necessarily offering predictive markers, but they're including or excluding based on genetics. What are the trade-offs in developing predictive markers, which would take a long time to develop and they would be costly and they might make the trial process go slowly? So what's the trade-off between those types of eligibility criteria versus developing predictive markers?
Yeah, this is a tough question. You're right. And I think, back to, what has been talked about today, I think some of the biggest successes we've had have been genetic, in small genetically defined patient populations. And that by nature, I think, excludes a lot of people. But it's shown how doing the right type of patient selection can really make an impact. And the challenge, which I fully understand, is in the ALS, there's a huge sense of urgency. And what that sense of urgency often equates to is everyone wants to be a part of a study. And I understand that dynamic, but then it's also has to be counterbalanced with, if you enroll everyone, you're also not doing the type of stratification that we've been talking about today to identify those patient subpopulations. And so, and because the metrics and the biomarkers we have are not very good right now, it becomes difficult to run any study where you exclude certain patients. And I understand why people don't like it. So I think I'm not sure there's an answer yet. But I think the first answer to me comes back to, what Michael and Corey have talked about, which is, we have observational studies, better biomarkers. And if we had a really good understanding, this is the subtype of disease that I might have versus another person, and there's confidence in that, hopefully then people can, feel better about how inclusion criteria and trials work. Because I will say, as therapeutic developers, we would love to treat everyone, right? But we want to give our medicines to the people where we think it's going to be most impactful for them. And that's where the balance lies. And we want to try to continue to get better at it. But definitely a challenge. No answer yet, but working towards it, hopefully soon.
Thank you so much. We have more of a general question about clinical trials. Michael, maybe you can address this. But Carolyn's asking if clinical trials can be administered in ALS clinics, and kind of paired with when people are already coming in with routine care. So I know a lot of trials do happen within the same clinics where people are going for care. Are there observational studies that can also fit into that paradigm well?
Yeah, so I want to be sure I understand the questions. I'll try to answer. Please come back if I don't get it right. I think that, a lot of research is done at centers where there are multidisciplinary clinics that are taking place. And certainly at our center, we try to minimize the burden of travel and all the associated burdens. So if somebody's coming to a clinic visit and they want at the same time to participate in an observational study or do a trial visit, we will do whatever we can to accommodate that. We also recognize, and we hear this from our patients, sometimes that's too much in one day. To be in the clinic all morning, seeing 6, 7, 8, 9 providers, even taking a lunch break and then spending 2 or 3 hours getting an infusion, going through assessments for a trial, it may be too much. So I guess for me, and I hope for patients too, we have to individualize this. We have to understand where somebody is coming from. If you're driving 3 hours, maybe it makes sense. Maybe a better plan is come and stay overnight, and the study will support you staying in a hotel overnight. You'll come to clinic, you'll rest, you'll do this trial visit the next day, and then you'll go home. Or if you live nearby, maybe it's easier to come back and forth. And so I think we have to try to tailor this to make it as easy as we possibly can, and reduce the burden for people to participate. So I hope I got at the question you were asking, but please come back if I didn't.
I think you did. And I think a lot of people have trouble finding research studies as well. And so we do have programs at North Star to help people kind of prioritize and find research studies. But yeah, there is a burden to traveling. And I think that you addressed it very well. There's also, it makes the day longer to incorporate those things into a clinic visit as well. And those days are very long. Um, all right. We have another question from Kevin about is trial design too focused on single drugs? Why not take moonshots on downstream issues by combining treatments? And we've seen a little bit of this in ALS, but I always find this a very interesting question, but it's hard. I struggle with understanding which drug is doing which thing. And so I'd love to hear Michael and Joe from the two of you on combining therapeutics in a trial setting.
You want to go first,
Michael,
Joe?
you go ahead.
All right. So I guess the answer is yes. No. Let me try to explain. I think it's almost certainly true that we've not done enough sort of combinatorial sort of therapeutic approaches in ALS. And so the way I sort of think about this, I often think of the analogy of sort of throwing a pebble into the pond and then there's sort of ripples that come out. And in this analogy, that pebble that hits the water is the cause of the disease or the upstream biology, and the ripples that come out are the downstream mechanisms. And I think what I imagine happens is that there's an amplification of disease as mechanisms kick in. Let's say you have neurodegeneration that's primary, and then you have an inflammatory response that's secondary to that. That inflammatory response may be beneficial, it may be harmful, but to the extent that it's harmful and it promotes further neurodegeneration, you sort of get into this sort of snowball effect. So I think what lies behind Kevin's question, as I understand it, is if we don't understand the upstream drivers biologically, but we see sort of all of these mechanisms that are active downstream, should we target many of them together? And I think there is some rationality to that. The one thing that I would just say is even when we see those many mechanisms, often we don't know what's protective and what's harmful and what's neutral. And so there's still a complexity in teasing out which of those multiple mechanisms we should target. But to come back to what I said initially, I think there's a general truism that there is sense when pathobiological mechanisms are synergistic in a bad way or additive in a bad way, that it makes sense to target multiple mechanisms at the same time. That raises its own set of challenges in drug development from a safety perspective and a tolerability perspective, and the potential for interaction and figuring out if something was bad or went wrong, what the driver of that was. But I agree with the general premise that we haven't done enough of this. But now I'll toss that football to you, Joe.
No, I think you described it well, Michael. And in principle, I love the idea of combinations. I think that's the most likely to be successful. In practice, it has its challenges, and I think Michael spoke to some of them. And most notably, it's every drug comes with complexities, and that's part of its activity. And if you add drugs together or add mechanisms together, you add all of those complexities together too. And the thought is, yes, if you add these mechanisms together, you're going to have a more likely chance of success. But what's hard to dissect is, does a particular drug, is it just not enough on its own and would work in a combination? Or it actually doesn't work at all and could be detrimental, right? And you kind of have to answer that question a little bit first, get some evidence that there's something to a drug on its own before you can bring it in combination with some others. And I think that's one of from an operational practicality perspective, that's one of the limitations is you need, even just to say from a mechanistic perspective, these are all working and they're safe to combine. I think then we can get there. So it's the dream. But I think it's going to take work and collaboration to get to a point where we can really run trials that allow that to be tested.
Thank you so much. Corey, I don't know if you have anything, I don't want to put you on the spot, but if you have anything to add from the oncology space, it would be great to hear your thoughts.
Yeah, it's interesting. I'm having these conversations in parallel with different oncologists thinking, in oncology, typically what you do is you treat with one drug at a time so you can attribute whatever the result is in order to get an FDA label for that one thing. And then that goes into to standard of care. And you end up in an advanced setting where you're treating one person with one drug, and then you wait until that person's cancer kind of blows up and gets out of control before you switch. And at that point, it's similar to it rhymes with what we were talking about earlier. You want to treat when there's minimal amounts of disease. And so we're now thinking about how do you is it better to just treat with combinations that you can try and understand the toxicity of in model systems so that you're like, to the points that were being made, that you're not just adding toxicity without any kind of benefit to the patient, and maybe not try to tease out what the attribution is, but rather index on trying to make the patient live longer? Or are you trying ultimately to get the drug approved for larger populations of people? And that's sort of what we're grappling with right now, is like treating the individual or carving the path so that you get approvals for larger populations of people. And I think those two things are a bit in tension.
Thank you so much, Corey. That was great. Michael, we have a question from Mark in the chat asking about diagnostic delays. I'm going to take us up to the last minute here, asking about diagnostic delays and what might we be able to do for earlier diagnosis so that people can get access to research and treatment earlier.
It's an excellent question and an intractable one. So I'll share some thoughts. I don't know that I have sort of magic solutions. I think just what's behind Mark's question is that, despite everything we've done, on average, the delay from symptom onset to diagnosis is about a year. And so that's way too long. I think the biggest reason for this is probably a health system issue rather than anything else. And health systems are hard to change. So what happens is somebody has a symptom that nobody, not the individual with the symptom nor the primary care doctor, recognizes might be an early symptom of ALS. And so what happens is we go down the wrong path. We get a referral to an orthopedic surgeon or a neurosurgeon for a carpal tunnel release or a lumbar surgery for a compressed root. And then symptoms continue to progress, and eventually someone says, well, perhaps go and see a neurologist. And then the neurologist says, well, perhaps go and see an ALS specialist. And so there's this sort of inefficiency of figuring out how to get somebody with a set of symptoms to the right person who can recognize the problem early. So that's part of the problem, is the health system problem. The other part of the problem, and we've sort of learned this in spades from our pre-FAL study where we're following people at genetic risk. For the longest time, I couldn't figure out what was going on because I was examining people, and I said, well, they're not completely healthy, but they don't have ALS. I'm like, what is this? Do I not know what this disease is? I can't tell these two apart. And then it dawned on us that we were observing a prodromal stage of disease that we've since called mild motor impairment, or MMI, by analogy to MCI in Alzheimer's disease. And it's the idea that there is a recognizable clinical syndrome that if we can get this into sort of the heads and the minds and the mindset of people at the front lines, the primary care doctor, the general practitioner, the general neurologist, maybe there's an opportunity to trigger earlier evaluation. But I will say this, the earlier we see people, the milder the symptoms, and the harder it is to be confident of a diagnosis. And it's in that context that we're going to need diagnostic biomarkers. And what I would say is right now, in terms of developing those, we're almost nowhere. And a big part of the problem is that we're not seeing people at that stage of disease where we can collect blood, collect spinal fluid, collect urine, and evaluate what sort of markers might be able to help us. So I guess what we need is a combination of changes in healthcare systems, changes in paradigms, and getting people recognizable clinical syndromes that they can recognize earlier. And we can see that that can work. In the Alzheimer's world, people recognize MCI. They don't wait for Alzheimer's disease. We need that in ALS. And once we can get to that stage, maybe we can move the needle on diagnostic biomarkers. So I think it's an excellent, critically important question, but the answers, I think, are layered and complex.
Really, really appreciate you. Juliana, you do have something to add there?
Yes, I wanted to chip in on the relevance of what Dr. Benatar was saying of studying people at risk. Again, this is a lesson we learned from the Alzheimer's disease world, where we followed a very large kindred with genetic mutation that would lead him to Alzheimer's disease. And it was only by following for over 30 years the same kindred that we were able to identify that the first changes in the abnormal protein deposition in the brain would happen up to 15 years before the onset of symptoms. This lesson learned from the Alzheimer's field should be definitely taken into the ALS world, where we are doing longitudinal follow-ups of individuals that are at risk, being for mutations on SOD1, C9orf, TARDBP, and trying to figure out if the first changes that we see in these individuals prior to really going into the mild motor impairment or full ALS, can this findings be extrapolated to individuals with not genetic or not monogenic ALS?
Yeah, Juliana, I think you're absolutely right. You know, when we first identified neurofilament rise in gene carriers, first criticism was, well, that's specific to SOD1. Turns out it's not true at all. People have gone back and looked at population studies like the Nurses' Health Study and neurofilament rises before this. We're now doing a study looking at MMI if we can identify it in community neurologists' offices, because it sort of goes without saying almost that before the disease is severe enough to be ALS, there must be something milder that comes before. This is not specific to genetics. This is going to be a general truism. So I think you're absolutely right. Sorry, Nadia, I know you need to wrap up. This panel could talk forever.
I know. I really appreciate you guys. I know we're over time, and I really appreciate that discussion because I don't think people always realize how the research from genetics carries over into everybody else. But thank you from the bottom of my heart for being here and for giving your time. We still have almost 40 people on speaking to how fascinating all of you are. We really appreciate your time and effort in working on ALS, and thank you so much to everybody who attended today.
Thanks for having us.
Yep. Thanks for having me.
Thanks for having me.
Thank you.
Take care.